Okay, good morning, afternoon, or evening, everybody. Welcome to the Bio-Optical In Situ Data Discovery and Access with SeaBASS webinar. This is your host, Jennifer Brennan. While everybody's logging in here, most of you have already noticed, so thank you for your feedback. We have two optional polls located at the bottom left and middle portion of your page. We appreciate the feedback we receive on these polls. It's 2 p.m. Eastern Standard Time, so let's go ahead and get started. First, what I'd like to do is review a few logistics related to the webinar. To ensure best audio experience, the conference has been placed in silent mode. However, if you have any issues or you have any questions, what I'd like for you to do is enter those into the Q&A pod, and you will see that located on the lower right-hand corner of your screen. This works like a chat. Today's webinar is being recorded. The recording will be posted both to our online Adobe Connect webinar catalog as well as to our NASA Earthdata YouTube channel within a couple of days of completion. And what I'll do is I'll send all registrants a link to the direct recording as well as a link to the YouTube recording as well. Today's presentation files will also be available for download at the end of the webinar. As far as timing is concerned, today's webinar is 1 hour long. We've allocated 45 minutes to the presentation and live demonstration with an additional 15 minutes for the question and answer period. So after our speaker has finished his presentation today, we will then transition to an optional final set of polling questions, and then from there we'll jump directly to the Q&A period. Again, you will have an opportunity to ask your questions throughout all portions of the webinar by using the Q&A pod. It works like a chat. Due to the large number of participants for today's webinar, we won't be answering any questions using the raising hand function. It has been disabled. And then just one final note, depending upon how many questions we receive, we'll extend the Q&A period an additional 15 minutes to 3:15 p.m. Eastern Time for those of you who may wish to stay on the line. What we'll do next is we'll pull up today's agenda. Give it just a second so everybody can see. Okay, during the first 10 minutes or so, our speaker will provide an introduction to ocean color remote sensing, and then from there he will introduce SeaBASS, both as an archive and as a bio-optical in situ data validation system. Finally, we'll wrap up today's webinar with information about algorithm development and the SeaBASS software analysis tool suite. With that, it is my pleasure to introduce today's speaker, Joel Scott, who is a scientific programmer and analyst at the NASA Goddard Space Flight Center, Ocean Biology Distributed Active Archive Center, or OB.DAAC. Joel?
Hi, thank you for that introduction, Jennifer. Just give me 1 second to get my slides pulled up. Again, it's my pleasure to be here today and to be able to talk to you a little bit about SeaBASS, which is a bio-optical data archive and validation system, as Jennifer introduced. Before we really get going, I'd like to give a shout out to my co-authors, Jeremy Bordell, Sean Bailey, Chris Proctor, and Ryan Vandermeulen, who all work with me here at NASA Goddard Space Flight Center in the Ocean Ecology Lab. Before we dive into details on what SeaBASS is and how it serves NASA and the ocean color community, I'd like to give you an introduction to OB.DAAC and SeaBASS. So OB.DAAC stands for the Ocean Biology Distributed Active Archive Center. Now this is NASA's center to archive and distribute satellite ocean biology data. The OB.DAAC also collects and processes ocean-related satellite products and is here to support the ocean color data user community. It does that through a couple different ways, and that one of those is through the data archive and distribution, but also providing tools to access and visualize that data. Additionally, the OB.DAAC serves to calibrate and validate operational and heritage remote sensing missions, and it is in support of NASA's EOS-DIS, which stands for the Earth Observing System Data and Information System. So OB.DAAC works with SeaBASS. We work hand in hand together. SeaBASS has a couple main objectives. First off, SeaBASS stands for the SeaWiFS Bio-Optical Archive and Storage System, and one of its main objectives is to archive and publicly host over 3,900 global in-situ bio-optical datasets. These data span from the 1970s to the present And all the data that is in the archive is plotted here on the map that you're seeing, the lower portion of the screen. A second objective of SeaBASS is to validate NASA-supported ocean color satellite products, as well as support satellite calibration and algorithm development efforts. Now, SeaBASS was originally founded in support of the SeaWiFS mission, which was launched in 1997. However, it has continued to serve other ocean color capable NASA-supported missions. The in-situ data that goes into the SeaBASS archive comes from a variety of sources. Ones that are worth mentioning are NASA's SymBIOS program, as well as NASA's Ocean Biology and Biogeochemistry program, and certain NASA Earth Venture projects, such as Coral and NAMES. We also accept voluntary submissions from the scientific community. So you might be sitting here wondering, well, what exactly is ocean color? Why do we care about it, and why are we remote sensing it? Well, let's start with the first part of that question. What is ocean color? You may have been taught as a child that the ocean is blue. Well, we all understand that the ocean is blue and water is blue. However, the ocean can come in a variety of different colors, whether it's more of a green hue or maybe a red or brownish color. Now, where's that coming from? Well, if you take a trip back to science class, when you learn the colors of the rainbow and how our eyes work, you may know the acronym Roy G. Biv, red, orange, yellow, green, blue, indigo, violet, the colors of the rainbow. Well, what we're seeing when we look at blue water is blue being strongly reflected from the sunlight to our eyes. So you can look at this in terms of which part of the rainbow spectrum is most strongly reflected. So for clear water, it most strongly reflects blue wavelengths. So you end up getting a spectrum of blue color coming to our eyes, whereas sediment-rich water that might appear more brown or reddish is going to have more of the red end of the spectrum coming to our eyes. So therefore, the spectral distribution of reflected sunlight to our eyes or to a satellite can be used to infer the contents of the water, whether it's clear blue water or whether it's sediment-rich water in perhaps a coastal area. So satellites work in a very similar way to our eyes, where they measure electromagnetic energy that's coming from the sun, transmitted through the atmosphere, strikes the surface, and then is reflected. So you end up having what's known as downwelling irradiance, photons coming from the sun, traveling through the air, and then striking the air-sea boundary layer. When that happens, there's two possible things that can happen. The photon can either be absorbed or it can be scattered. Now, what's doing that absorbing or scattering? Well, it can be the water itself. It can be organic matter, detritus, phytoplankton, all those constituents that make up the water. So light that is scattered away from that water's surface is known as water-leaving radiance. That is what our eyes see. That's also what the satellite sees. So what causes these variation of color in the ocean? Well, here's an example of water ranging everywhere from red to blue, and you can see how the water-leaving radiance profiles for each of those images is skewed to a different part of the spectrum, whether it's more towards blue or whether it's more strongly reflecting in the reddish end of the spectrum. The color of the ocean is a function of the light that is absorbed or scattered based on what's in that water. Now that can come from a number of things, whether it's phytoplankton, which are the building blocks of the ocean food chain, and the pigments that they have to conduct photosynthesis. It can be from dissolved organic matter, it can be from detritus or inorganic particles, and then also it can come from the water absorption itself, which water absorbs strongly in red and reflects more strongly in blue. So how do we derive what's known as ocean color products from space? Well, you have a satellite that's orbiting the Earth and looking down at the surface. So the satellite's viewing the spectral light field from the top of the atmosphere. So the first step in trying to get at ocean color products and trying to understand what's in that water from space is to remove the atmosphere from the total signal to derive the light emitting from the surface, which we call remote sensing reflectance. The second step is to relate that remote sensing reflectance to a geophysical product that we're interested in. In this example here that you're seeing, it's phytoplankton, and we're trying to get at the chlorophyll-a concentration. Well, that's where SeaBASS comes into play. So SeaBASS is a database of remote sensing reflectance data, as well as chlorophyll-a concentrations that have been measured in the field, and then we can relate those two together to derive an algorithm that can be applied to the satellite and what it's seeing. So when I say ocean color satellite data products, what am I referring to? Well, there's a couple of them that OB.DAAC supports, and I'll cover those just briefly and what they're getting at. Now this is an example here of chlorophyll-a, which gets at algal biomass. The region you're looking at is the Chesapeake Bay. Us being here at NASA Goddard, the Chesapeake Bay is very close to our backyards, and we love any opportunity to study the very interesting waters that occur there. Another ocean color satellite data product is particle backscattering, which gets at sediment load of the water. There's also diffuse light attenuation, which can give you an idea of water clarity or turbidity, as well as dissolved organic matter absorption, which gets at runoff and things that are dissolved into the water that then flows out to the bay. Of course, there are many other ocean color satellite data products that OB.DAAC supports. However, I'm just giving you a general idea of what I mean when I say ocean color satellite data products here. So there are a few NASA-supported ocean color satellite missions. As I mentioned earlier, SeaBASS was originally founded to support SeaWiFS, which is no longer on orbit. It's a heritage mission. However, there is MODIS Terra and MODIS Aqua, as well as the NOAA-NASA partnership of VIIRS, Suomi-NPP. And there's other heritage missions that we continue to support, such as CCCS, OCTS, and MERIS. So with that under our belts, let's dive right into SeaBASS and how it serves as a bio-optical data archive. So let's first define what exactly bio-optical data is. Well, there's a couple different types of data that can all be classified as bio-optical. And I have those listed here, but I'll elaborate on each one of them a little bit. So the first one's apparent optical properties. Now that's when you look at water, what do you see? So it's things like remote sensing reflectance, irradiance, all of those properties that are apparent about water. There's also inherent optical properties, which that's things like absorption and scattering coefficients. There's also phytoplankton pigments, where you're trying to understand what pigments the phytoplankton have and how that affects the color of the water. There's also carbon stocks and hydrography with currents and temperature and salinity information, as well as other biogeochemical and a diverse array of atmospheric measurements that are all relevant to trying to get at ocean color. Now there's a variety of different collection platforms that are used to observe these type of data and that are then submitted to SeaBASS. So you can have ships and the instruments connected to their flow-through system. You can have ships doing casts where they send out a profile or collect bottle samples at various depths. You have divers, moorings, drifting buoys, autonomous underwater vehicles, or different types of airborne deployments of instruments that can get at all of this type of data. So how does that data get to SeaBASS? Well, generally there's a NASA-funded project that will often require the in-situ field data collected underneath it to be archived in SeaBASS. We also accept voluntary data submissions, but the majority of our data is coming from NASA-funded projects. So once a project is funded and you have a fieldwork campaign funded, there's usually a deadline set, something like 1 year or so from the time that they're deployed and they collect that data, that the investigators can then run their data post-processing and their quality filtering and quality assessment and reformat that data and then submit it to SeaBASS. Once that data is submitted to us, eventually it will be archived in SeaBASS and made publicly available. But before that happens, there's a few format verification and documentation verification steps that we conduct on that data submission to us. We want to make sure that we're documenting where the data was collected, how it was collected, what exactly was collected, and all that relevant information. Once all those verifications are all set, the data then is archived into SeaBASS and made publicly available. There's also another step, and that is to generate satellite validation and algorithm development data sets that SeaBASS supports. And in order to do that, once the data is in the archive, there's further quality control and filtering that's run and post-processing done here at NASA before it's then matched up with satellite data to validate what the satellite's seeing or to be paired into an algorithm development data set. Now these last two boxes are two of the key ways that SeaBASS serves NASA and the ocean color community. So as I mentioned, there is some formatting that has to happen from when an investigator collects data and when it gets submitted to us. So SeaBASS is built on a SeaBASS data format, which is an ASCII file format, and it is one of NASA's Earth Science Data and Information Systems ESDIS approved data formatting standards. Now the SeaBASS format is broken into two different sections. There's the metadata header section, which is a series of keyword value pairs that describes information about who, what, when, where, and why, and how the data were collected. This gets at information on who the investigators were, what their affiliation is, how to contact them if you have questions, what experiment or larger funding program the data may have been a part of and was collected under, as well as what deployment and what cruise report information may have been accompanied that data, as well as relevant documentation and calibration files and protocols that were followed in collecting this type of data. The header section also includes information about your date and time bounds and your latitude and longitude bounds. This is all very nice and machine readable, so it's easy to ingest for us to index and make the data very searchable, as well as makes it easy to read into programming languages to further manipulate the data. I also want to point out that there's fields and units declared in the metadata header section, and these correspond to each column of data in the data matrix, which is the second part of the file. So the second part of the file is values separated by a delimiter. This is where the raw data is stored, and each column has a corresponding field and unit. Now, as I mentioned, with each of those columns having a field and unit, that's coming from a standardized list that we maintain at SeaBASS on our website. Here's just a small section of it, but the reason that we base this off of a standardized list is because there's a variety of ways to measure the same quantity, yet not all of them are created equal, so we want to be able to distinguish between maybe a chlorophyll that was derived fluorometrically or spectrophotometrically versus a chlorophyll that may have been measured through high-performance liquid chromatography analysis. In order to do that, as you can see here, just first two examples in this table, there's CHL versus CHLA, and that way it makes it easy for when you're ready to search for data or to find data, you can search specifically for various types of data that were derived in a similar way. Now, we curate this list and add new fields as necessary, and so the most up-to-date list of units and standard field names that can be submitted to SeaBASS is maintained on this URL here. We also sometimes have to add new metadata headers depending on what type of data is being reported, and so we also curate a list of the different metadata headers that are accepted and how they can be best used. So let's do a little example of how one might search for data in SeaBASS. If you were to go to the URL that you see here at the top of the page, seabass.gsfc.nasa.gov, you'll see this page. This is our home page. It's where you would start, and if you're wanting to just search for bio-optical data, you would start by clicking on the File Search icon. Once you do that, you'll see a page very similar to this one where you're allowed to input search parameters for the data that you're interested in. You can specify when the data was measured between or when it was archived in SeaBASS between. You can also specify latitude and longitude ranges that you're interested in, or if you don't exactly know the exact latitude and longitude, you can use the interactive map to draw a region that you are interested in searching for data from. Additionally, you can provide specific search keywords such as your affiliation, the investigator who took the measurements, the experiment program that it was a part of, excuse me, as well as the cruise deployment that it may have been a member of. Scrolling further down the page, or in this case next to it, you can also specify different products. We can pick a grouped product, or you can enter a specific product name, and that's coming from that standard field list again. Now, there's a nice feature here that you can search for files that contain any of the selected products or product groups. You can also search for data products that were measured within the same cruise, so that way you can get at contemporaneous data in space and time. Once you have your search performed, you'll end up getting a results page similar to this one. Now, we try to make the results page as intuitive and provide as much information we can as possible on it, such that before you download any data, you can preview what you're getting and what your search has returned. First feature I would like to highlight is the ability to map the data. Clicking on the Map All button, you'll end up getting a map like this that shows all of the locations of the data that your search returned. We also provide bulk downloading or downloading selections of data, and that will include different calibration reports and associated files and cruise reports. You can download all of that data by clicking the Download All button, or you can use the Download Selection tab. We also believe that you should be able to plot the individual data that's contained in the files just in case you want to preview a file before you actually download it. So clicking the plot icon, you end up getting an overlay like this that comes up where you're allowed to select your X and your Y parameters, or your Z if you have one, and then you end up seeing a plot very similar to this one, which here we're looking at remote sensing reflectance versus wavelengths. We're looking at a spectral distribution. Now, this data itself was measured by Zhongping Li under the Coral project in the Great Barrier Reef near Heron Island. Now, as I mentioned, you can also look at data in 3 axes. So if you have data that was spatially collected, such as versus longitude and latitude, and in this case chlorophyll, you can also choose those options, in which case you would get a color plot like this, where the color corresponds to the chlorophyll concentration that was being reported in that file. So again, all of these are features that you can access about the data that's archived in SeaBASS before even downloading anything. So you can preview the data, see what's there, and see what you're getting. Additionally, some of our data can have associated files such as images or other types of data bundles. If that's the case and there's a preview available, you'll end up seeing an icon like this. Clicking that icon ends up showing you a preview of the associated imagery. Now in this specific case, we're looking here at a benthic cover photo mosaic. So this is where divers swam along and took high-resolution imagery of the ocean seafloor and the benthic cover that was there. These files themselves can be up to a number of gigabytes, so we downsample those and provide previews so that it's much more efficient. Instead of you having to download massive amounts of imagery, you can just click the preview and see the associated image if you're interested in downloading it. Additionally, every data set that is submitted to SeaBASS is assigned a DOI, which stands for a Digital Object Identifier. And these are useful, one, to link data together, but also to cite data. With a DOI, you end up getting a permanent web page, which we host on our SeaBASS website. Here's an example for the Coral campaign. And you can see this web page screenshot shows the different investigators that collected data, the dates and times of the data collection, and as well as the latitude and longitude bounds and the different parameters and data types that were measured. You also see there's a brief description provided and relevant URL links are provided as well. Now this is useful because many journals are now moving to a requirement where you have to have your data cited in addition to your normal journal references. So if you were to use that benthic cover image that I just showed on the last slide, you would end up needing to include a citation like this referring to Eric Hochberg and the data that he has archived with us in SeaBASS. So SeaBASS very much values serving the scientific community, whether that's the ocean color community or the modeling community and the general circulation models that have bio-optical components that assimilate our data, as well as aerosol and atmospheric researchers who might be trying to tackle that problem of ocean color retrievals. And we've also even worked with satellite engineers and designers to help set new standards for new instruments that are upcoming and that are being developed by NASA. Of course, there's many other users that we have. I'm just shouting out to a few here. But let's move on to SeaBASS as a validation system. So as we just covered, you end up having observing platforms that are out there at the ocean, such as in this case we use a diagram of a boat, that are making measurements of the ocean and the sea surface and the constituents in the water, and then sending that data back to SeaBASS to be archived. So that's how SeaBASS serves the ocean color community as a bio-optical archive. But you also have satellites orbiting the Earth, viewing those same scenes as the boat. This gets at how SeaBASS can serve as a satellite validation system. The goal here is to pair in-situ data in time and space to the values that the satellite's seeing. Now, in order to do this, there's a couple in-situ data post-processing steps that have to take place. The first step is to do a quality assessment and filtering on the in-situ data. We want to make sure that the data that we're going to be using to validate our satellite retrievals is of the most high quality and doesn't have any erroneous measurements or blips from instrument error or any of that. Additionally, we verify all the protocols that the data was collected under just to make sure that the measurements and the types of data measurements that we're getting at actually makes logical sense that how they were collected and there was a rigorous protocol applied for the measurements that were taken. Additionally, we adjust the values that are in situ to water-leaving values. Much of the data we receive at SeaBASS is coming from a cast or a profile where it was taken at a specific depth, whether 10 meters or 30 meters down in the ocean. But you can't take that measurement and compare it directly to what the satellite's seeing. That's like comparing apples to oranges. So fortunately, there's ways to model the properties of the water column and be able to adjust those measurements that were taken at a certain depth to the surface such that it can then be compared to what the satellite's seeing. So let's go through a little example here on how you would use SeaBASS to search for validation data. You would again start at the SeaBASS homepage, but this time you would click the validation search. You would end up seeing a page very similar to this with the same kind of options as before. However, scrolling further down, you'll see a different set of options where you are allowed to select the satellite sensor that you're interested in. And then you can also select different satellite filtering criteria or exclusion criteria. And that gets at the time difference between the satellite overpass and when the in-situ data was collected, or different information about the viewing geometry of the satellite, whether it's looking off to the side or right underneath the satellite track, or different solar angles or any of those different types of things. However, if you just want to take the defaults that we provide, those are coming from the Bailey and Wardell 2006 paper, which was a rigorous study to try to understand the best way to compare satellite data to in situ data for validation purposes. Additionally, all the satellite data that we are providing validation and matchup results for has already been quality filtered in terms of making sure it's not over land, that there's no sun glint contamination or stray light, to make sure that there's no cloud or ice contamination in the satellite retrieval, and also to make sure that the satellite is looking where we think it is and that there's not a navigation or a geolocation failure. Finally, you would select what product you're interested in to validate from the list there, and then you would click Submit, and you would end up getting a page that looks very much like this. Now what we're looking at here is chlorophyll-A is what I chose, and on the x-axis you have your in situ data, and on the y-axis you have MODIS Aqua. There's a table of summary statistics at the top with your product name, the number of matchup points you have, as well as some various goodness of fit statistics from mean bias, mean absolute error, random error. Now what we're seeing here is that over the time period that MODIS Aqua has been flying and the 951 in situ validation points that match up, has relatively good agreement, which is useful to NASA to know that the algorithms they have implemented and that are being run have relatively good agreement with the in situ data that have been collected. However, this is looking at everything through all time that MODIS has been up there that matches with SeaBASS's in situ data records. However, sometimes it's useful to look at things through time in order to see how stable a satellite instrument is or how a certain region is changing in time. So I have one example here, and this is a newer feature that we're adding to SeaBASS in the coming months. But this specific example here is of the Chesapeake Bay again. We broke it into 3 different regions where there's an upper bay, a middle bay, and a lower bay, as you can see by the yellow, blue, and green color-coded areas in that chlorophyll-A image there. Now the circles and the shaded areas represent in situ chlorophyll measurements. The blue line is the SeaWiFS time series, and the red line is the MODIS Aqua time series. And you can see how there's agreement between the in situ data or not agreement between this in situ and satellite data through time. Now, ideally, we would like to be doing this everywhere, all the time. However, I broke out our validation data into years, and so here you have 4 panels, and just going clockwise, there's 2006, 2007, 2008, and 2009. And you can see, just looking at an individual year, the data in SeaBASS for validation is rather sparse. Ideally, we would like to be performing validation matchups with lots of dense in situ coverage for every year. However, we have to stick to regions to do the validation time series type analysis. However, we are working with autonomous data sources, which have their own set of challenges and complications. One of the main ones we work with is AeroNet, the AeroNet network, which is a fixed above-water platforms that take radiometry information as well as ocean color information and then report that back in an autonomous sense. We also collect data from different buoy networks that operate fairly autonomously, as well as gliders and drifters, which can be deployed for weeks or months at a time, as well as towed or underway sampling from different ships of opportunity. However, with autonomous data sources, you have to be more careful with your quality filtering and make sure that your instrument hasn't degraded since it was deployed. So moving on into how SeaBASS supports algorithm development efforts. So we just finished covering how SeaBASS works to validate, which is where you're comparing in situ data to satellite data. However, for algorithm development, we're really interested in pairing different in situ data types together. So in this kind of situation, you would want to compare IOPs, which are your inherent optical properties, your absorption and your scattering coefficients, and the pigments of that water samples that were collected. And you want to pair that with your radiometry or your apparent optical properties. So what the water looks like. Frequently, these types of measurements are taken during the same cruise. However, they might have been by a different investigator, or they could be stored in separate files in SeaBASS. So what we're doing here through our work to provide algorithm development data sets is to take different data types and pair them in one common data set and data file such that it's easy to relate these two things together, so that researchers can come to us and access that algorithm development data set, which already has different data types co-located in time and space, and then they can relate them together to develop a new algorithm. Once that algorithm is developed, you can then apply it to the satellite in order to develop new algorithms that can be implemented on the satellite data. So SeaBASS supports all those efforts through the NASA Bio-Optical Marine Algorithm Dataset, or NOMAD as an acronym. NOMAD is a highly quality controlled dataset, again, designed for algorithm development. It contains co-located water-leaving values of those AOPs, the radiometry, the IOPs, the absorption and scattering properties of water, as well as different pigments, so that all of these can be related together to develop new algorithms. Now, all of these data are spatially contemporaneous, and additionally, they're holistically evaluated, which means you're making sure that the pigments make sense for what you're seeing in the radiometry, and all of the data makes sense on whole for a specific data point. Now, as I've covered already, when you get to this level of high-quality data, you end up dropping off a lot of that data that's archived in SeaBASS. So the map here you can see is a little bit sparser than our entire data records, but it covers these regions with very highly quality controlled and contemporaneous AOPs, IOPs, and pigment data. So again, the NOMAD dataset can be accessed from our main website just by going to SeaBASS's homepage and then clicking on the NOMAD dataset. And you'll find a lot of great information there on NOMAD, how it's curated, how it's developed, and then also how to access it. So we have all of this data and we have this SeaBASS format. We also try to provide software and analytical tools that helps one, read and access the data that's stored in that format, as well as convert it to different formats. So for the SeaBASS file format, we provide readers in MATLAB, Python, and Perl, as well as tools to write SeaBASS files in Perl and Python. Because the last thing a data user wants to see is, oh, there's some other new format that I have to write a reader to get it into my favorite programming language. So we developed these tools to try to take out that barrier and make it as easy as possible to use the data that one downloads from SeaBASS, whether it's a validation file or the NOMAD dataset or just a simple Bio-Optical file. We also provide file converters because perhaps you're more comfortable with NetCDF or an iCart format, in which case we provide web tools for you to convert NetCDF data, convert SeaBASS files to NetCDF or iCart format. Also, if NASA ran your high-performance liquid chromatography pigment analysis, the report files that they generate and send to you, we provide a converter that can directly convert those spreadsheets to SeaBASS files. Also, sometimes we have users who would like to conduct their own satellite validation matchups in addition or outside of what we're providing through our validation search tools. And so we offer two scripts to do that. The first script is a satellite data locator and download script. You give it a SeaBASS file, it goes through and extracts the date, time, and latitude, longitude locations, and then it conducts Earthdata searches to figure out which satellite data was coincident during that period, and then it can download it to your local machine. The second script then takes that downloaded data and can extract, filter, and pair the satellite data with the in-situ observations, storing it all out as a SeaBASS file, which can then be read with those reader tools in MATLAB, Python, and Perl. Again, all of these tools are available from the URL you see there on the screen, and they'll have brief description pages that explains how to access them, how to download them, and how to use them. We also provide a FCheck file checker. Now this is more for our data submitters who are trying to format data into SeaBASS files. And now this FCheck file checker verifies the SeaBASS file formatting, the metadata header used, the fields and units, the data ranges, and it provides feedback on how to correct data files that might have incorrect formatting or perhaps some suspect data that's outside of the expected ranges for certain variables. Our goal here is to try to provide as intuitive a feedback as possible so that it makes it easy for submitters to us as well as us to review data and get it into the archive. There's three different ways to run FCheck, and that you can email files as attachments to FCheck to be automatically checked. You can also upload data to our FTP server. If you have a bulk data, 500 or more files, you can easily upload them all quickly and then run FCheck on all of them in bulk. Or if you prefer a more advanced approach, you could download the source code for FCheck and run it locally on your machine. So in summary, SeaBASS supports NASA and the ocean color community with a few key deliverables. There's the Bio-Optical in-situ data archive with its powerful tools to index different bio-optical data, provide search tools to map and visualize the data and bulk download the data, as well as providing resources to cite that data that you may use. There's also the satellite product validation system where you're taking in-situ data, adjusting it to water-leaving values, and then comparing it to NASA-supported satellite products to see how the satellite algorithms are performing and ground truth that information. There's also the NOMAD algorithm development data set where you're pairing different data types from in-situ observations together so that you can develop new algorithms. And then finally, there's software and data access tools that we develop to make it easy to use the data and access what we are hosting here at SeaBASS. So I'd like to thank you for your attention and leave you with these links to SeaBASS and the OB.DAAC's webpage. And I'd also like to thank my co-authors and acknowledge and provide acknowledgments for image credits to Javier Concha, Norman Curring, and the Ocean Biology Processing Group at NASA Goddard Space Flight Center. Thank you.
Okay, well, thank you, Joel. Thanks, everybody. At this point, what we're going to do is we're going to switch it over to a final set of polling questions, and we'll give these questions about 3 minutes or so since we finished a little bit early. And then from there, we'll move directly to the Q&A period. Although you don't see your questions now, for those of you who have entered questions already, thank you. Not to worry, the Q&A log is a persistent feature, so just as soon as we finish up with these polling questions, we'll transition directly to the Q&A, and we'll get started with that. All right, well, thanks, everybody. Okay, everybody, we're going to give these questions just about another minute and a half, and I just want to tell you that we really appreciate the feedback on these questions, particularly for future webinar topics. It really helps to, create our webinar schedule over the next year. All right, so thank you. We'll give this another minute or so, minute and a half, and then we'll move directly to the Q&A period. All right, everybody. At this point, we are going to go ahead and transition to the Q&A period. And if you think of something that you'd like to ask, feel free to jot it down as well within the Q&A pod. That's also an option if you thought of something after we, kind of moved on. So our first question was submitted a bit ago, and if you give me just a moment to pull that up. Here. Okay. The first question is, are there required protocols for collection and processing, for example, above water, handheld, hyperspectral radiometers? So are there required protocols for collection and processing, for example, above water, handheld, hyperspectral radiometers, ASD, etc.?
That is a great question. Thank you for that. We at SeaBASS do not enforce any required protocols. There are certain protocols that NASA recommends, and also NASA has a number of protocol development activities. I can put you in touch with that information if you would like to send me an email with the people who work on that. However, at SeaBASS, we don't have any explicitly protocol requirements.
Okay, thank you, Joel. The next question is, what level of satellite data, level 2, level 3, is used in the validation search feature?
So in the validation search feature, we're actually, because we're here co-located with the OB.DAAC, we actually run custom processing from level 1 to level 2 to generate those products. But to answer your question, the closest level we're running validation on would be level 2 products. So for those that are not quite sure what that means, level 2 would be swath-type geophysical retrievals, whereas level 3 would be ones that have been subsampled to more of a map-type orientation.
Okay, thank you, Joel. The questions are rolling in here now at this point. Okay, so the next question, let's see, bear with me one moment here. How many pixels are extracted from the satellite data for matchup analyses?
Generally, that's covered in the Bailey and Wardell paper of 2006, but generally it's somewhere between 3x3 or 5x5 type pixel areas that's extracted from the satellite data, and then there's also, like I mentioned on the exclusion criteria, you can specify how many of those pixels are valid and pass the different flagging criteria, and we're doing that. All that logic can be covered quite thoroughly in the Bailey and Wardell paper. But it's, again, just trying to get at making sure that you're comparing apples to apples for what the in-situ data is and what the satellite's seeing.
Okay, great. Thank you. So the next question is, okay, would SeaBASS be interested in CTD fluorescence data?
Yes, that sounds like something right up our alley. Yes, very much so.
Okay, and so I should say for this particular user, if you don't already have the contact information for our speaker today, I don't know, do you have is there a help email address that I can type into the Q&A pod, Joel? Or would it be preferable to follow up offline directly?
Follow up to offline directly. I think my email address directly is great to get in touch with me. There's only a handful of us that work at the SeaBASS Data Archive. And I can get you in touch with the right people.
Okay, so I will send that information along after the webinar. All right, and the next question is, there was no in-situ ocean data available in southern Alaska, northern BC Canadian coast. Is there a reason for that?
Not to my knowledge. There's not a reason other than just no one has sampled that area. It's the data that we have is either NASA-funded projects or some of those other programs that I covered. And I don't have a specific answer, but I'm guessing no one has collected data there. There's not a specific reason we're not blacklisting it or anything like that.
Okay, thank you. The next question is, and also a comment, thanking you for the lecture. What do you personally think, Joel, about the future of this system in the next 5 to 10 years?
Well, I care a great deal about all of this data. I'm very passionate about it, so I would love to think that it continues on for the next 5 to 10 years. I think it's a great resource. It has data spanning a number of different satellites and supporting different validation activities as well as algorithm development. We're always looking for new ideas on how we can serve our user community and make our data more useful. So we're trying to make those database search queries and things like that more powerful, more useful. And we welcome feedback that any users may have.
Okay, great. Thank you, Joel. The next question is, what upcoming satellites are planned to be added in the future?
That is a great question. Unfortunately, I don't have a fortune teller's ball in front of me. There is the new VIIRS that was recently launched, but we haven't been provided guidance to support that yet at OB.DAAC. So when that happens, I'm sure there'll be announcements about that. If it does happen, we don't know. So just stay tuned.
Okay, thank you. The next question is, can SeaBASS be used to massively download regridded satellite data?
Actually, that would best be handled through OB.DAAC and its resources. There's a number of I think you'd be interested in level 3 satellite data. And there's a level 3 data browser that you can search for specific data. That's more at the OB.DAAC side, so just follow the OB.DAAC URL that I linked to at the end of my talk, and you can be able to find that there. We don't provide any massive gridded satellite data through SeaBASS, however.
Okay, thank you, Joel. And what I've done is I've also added the, the ocean color, the main link to the Q&A pod as well. And in that vein, while we're waiting for additional questions, you'll notice in the bottom right-hand corner, the presentation files for today as well as the agenda are posted and available for you to download if you're interested. What you'll do is you'll click on either one of those 2 files and be prompted with an option to download the file. Are there additional questions? Anybody? Yes, there's an additional question. Make sure I haven't forgotten any in the, I'm looking through. Okay. The next question is, are chlorophyll product algorithms general or regionally tuned?
It really depends on which algorithm you're looking at. Again, it's a better question for the OB.DAAC side of things. There's a number of documents that describe the algorithms that are run on the different satellites. Again, and all that information is accessible under the documentation tab on the OB.DAAC website.
Okay, thank you, Joel. Let's see here if we have any additional questions. Does anybody else have any additional questions? Don't think I've missed any. Just doing another scan through. Okay. The next, it's more of a comment and an inquiry here. So the next comment/question is, it is important to maintain ability to match up user-supplied or SeaBASS-supplied latitude, longitude, time, in-situ points to satellite RRS, not just a specific satellite product.
I agree with that. We do provide matchups for RRS, remote sensing reflectance. It's not just a specific product. It's not just specific NASA-supported satellite ocean color products, but we also do provide those matchups for RRS as well. Again, under the validation search on the SeaBASS website.
Okay, thank you, Joel. Excuse me. And then, let's see here. Okay, the user is indicating that the last time he or she tried that, it was a bit involved in trying to get to RRS.
I believe you just go to the validation search page. After you selected which satellite instrument you're in, you would scroll down to the bottom where you can pick your products, and you would check the RRS box, and then you would click Perform Validation Search. You would then get a result panel very similar to that scatter plot that I showed for chlorophyll. However, it would be for each of the RRS channels for the relevant satellite sensor. Also from the validation search results page, you can click on the data tab, and it will actually give you a SeaBASS file of all of the data that goes into making those analytical plots that you see on the validation search results page. If you would like, please send me a direct email if you are having any issues, and I can try to work with you to figure out how to best resolve those.
Okay, thank you, Joel. And I will provide the Q&A log to our speaker, so he'll be able to follow up offline very easily for any additional questions that might require, a direct interaction. So no worries on that one. Are there any additional questions? If there are no additional questions, here's what we'll do. We will log off from the audio component for this webinar, and then we will leave the virtual meeting space open an additional 10 minutes. We finished a bit early. Thank you for the kind words, Cesar. That's good to hear. I'm glad that people have enjoyed today's webinar. So we'll leave the meeting room open an additional 10 minutes for those of you who think of something you'd like to ask, or you're interested in downloading today's presentation files. And then just again as a final reminder, particularly for those of you who may have joined a bit late, if you're a repeat attendee, you'll know that I will send a link to the direct recording as well as to the YouTube location for today's webinar within a couple of days. There's a bit of post-processing involved, and so be looking for that. And with that, I'm not seeing any additional questions. So I don't know, Joel, if you have any final remarks. If not, we will log off from the audio component, and from there we will leave this virtual space opened.
I would just like to say thank you to everyone for attending, and I hope it's very useful to you. Thank you.
Well, I do have one more. My apologies. There's one more question. Okay, so the question is, this particular user is wondering if it's possible to change the atmospheric correction models for validation purposes.
Not currently. However, with the tools that OB.DAAC provides through SeaBASS, which there was another webinar given a little while back on that, one can apply different atmospheric corrections to the satellite data, and then using those standalone validation matchup tools that I covered in the software section, you could then create those matchups to your in-situ data that way. So there is a way you can do it, but it just requires a few extra steps. It's not something we currently provide through our validation search page.
Okay, thank you, Joel. All right, let me just scroll down here. Okay, there's another, for matchups. Okay, for matchups, how do you deal with the time resolution of satellite data? Do you time average?
Generally what we do is we set like a time window, and that can be specified under the satellite filtering options. The default, I believe, is a 3-hour window. So again, we're not averaging the satellite data in time, but we are looking for the nearest observation within a time window.
Okay, thank you, Joel. Let me make sure I've not missed anything here. Just give me 1 second. Okay, any more questions? Last call for questions. Okay, if there are no additional questions, then we will log off from the audio component, and I'll leave the virtual meeting space open. And thanks to all of you for your very valuable input, not only for the polling questions and during the Q&A. It's extremely helpful as we plan our future online events. I'd like to thank all of you. Thanks for taking time out of your day for attending today's webinar. We have one next week on Tuesday, the 30th, which will focus on the state of the ocean data access and visualization tool for physical oceanography data at our physical oceanography DAAC. So this month is ocean data focused. All right, well, thanks everybody. Thank you, Joel. We'll log off from the audio component at this point. Okay, goodbye everybody.